Hello students, how so you are doing well. And in the previous lecture of protein we have discussed about the structure of amino acid, function and structure of proteins and importance of proteins. And in this lecture we will discuss about the structure of protein and their levels of folding, primary, secondary, tertiary and quaternary. And after that we will discuss about the classification of proteins, that is the globular and that is globular and fibrous proteins. So in the first part of this lecture we will discuss about the structure of protein. And structure of proteins actually, uh, because of the number and specific sequence and the foldings or bonds that are present in the proteins may vary in different proteins. So we can see primary structure. Actually primary structure is actually simple. There is a no band, there is no folding, only the number and sequence of amino acid is present in the primary structure, and only one type of bond, that is the peptide bond, is present in the first degree of or we can say primary structure of the proteins. Actually here two examples are given which are just describing the number of amino acid and number of peptide bonds in the protein. So we can say there is, after the F. Sanger's work of 10 years, it was concluded that insulin composed of 51 amino acids in two chains. There is one chain alpha and second is beta chain, and total 51 amino acids are present. And there are 21 amino acids that are present in the one alpha chain, and there are 30 amino acids that are present in the beta chains. So one of the chains, you can say that totally insulin is composed of two chains and 51 amino acids. One is alpha, one is beta chain. Alpha contains 21 amino acids and beta contains 30 amino acids. And if we talk about the peptide bond that are present in the insulin, total peptide bond will be the 49, because uh, total amino acid that are present in the alpha chain are 21. So of course definitely at the first and at the last amino acid there is a no peptide bond. So you can say that there are 20 peptide bonds in alpha chain and 29 peptide bond that are present in the beta chain. So there are total 49 peptide bond that are present in the insulin structure. And after that, hemoglobin is composed of four chains, two alpha and two beta chains. So there is a trick for the students, that if any insulin, if the number of chains are present, if there are two chains in any protein, the difference of the number of amino acids and peptide bond will be the that shows, if you are supposed to think that insulin has two amino acids, sorry, two there are two chains in which 51 amino acids are present, and of course because of difference 51 minus 2 49 peptide bond will be there. And if any protein has four chains, then you can say the difference of the total amino acid and number of chain is equal to peptide bond. And if we are saying 574 amino acid that are present in the hemoglobin in the form of four chains, and there will be 570 peptide bonds. And four chains, two alpha and two beta chains, each beta contain 141 amino acids and each alpha contain 141 and each beta contain 146 amino acids. And let's start with the reading of the primary structure. The primary structure comprises the number and sequence of amino acids in protein molecule. F. Sanger was first scientist who determined the sequence of amino acid in a protein molecule. After 10 years of careful work, he concluded that insulin is composed of 51 amino acids in the two chains. One of the chain had 21 amino acids and the other had 30 amino acids, and They were held together by disulfide bridges. Hemoglobin is composed of 4 chains, 2 alpha, 2 beta, and each alpha contain 141 and each beta contain 146. The size of protein molecule is determined by the type of amino acid and the number of amino acid comprising their particular proteins. So we have discussed the primary structure, and there is a one example given. The specific sequence of amino acid is determined by actually the specific nucleotide that are present in the DNA, and if any sequence is missing or if any one amino acid is missing due to the change in sequence of the DNA. So there is one example. Sickle cell hemoglobin is the one example in which the only in each beta chain at carbon number 6 one amino acid is replaced by another amino acid. We can say in normal hemoglobin on carbon number 6 of each beta chains the normal amino acid is glutamic acid, while in the sickle cell anemia this glutamic is replaced by, sorry, in the normal it is a valine, and in the abnormal it is the glutamic acid. So you can say that valine is replaced by the glutamic acid at carbon number 6. The best example is sickle cell hemoglobin of human being. In this case, only one amino acid in each beta chain out of 574 amino acids do not occupy the normal place in the protein. Actually, that is the valine in normal and glutamic acid in abnormal. Valine is replaced by glutamic acid. In fact, this particular amino acid is replaced by some other amino acid, and this is the example given. And in the next structure, that is a secondary structure, first of all we will discuss about the bonds. There are two type of bond: peptide bond that are present in all the structure of proteins, that are primary, secondary, tertiary and quaternary, while addition is one hydrogen bond. So it is, there are two examples actually. There will be the bend in the structure of protein and it is called the alpha helix and the beta plated sheets. These are examples of the secondary structure amino acid, and in each turn there are 3.6 amino acids in each turn of helix. So let's start. There is all about secondary structure. The polypeptide chain in protein molecule usually do not lie flat. They usually coil into helix or into some other regular configuration. One of the most common secondary structure is the alpha helix. It involves spiral formation of basic polypeptide chain. The alpha helix is very uniform geometric structure with 3.6 amino acid in each turn of helix. The helical structure is kept by formation of hydrogen bonds among amino acid molecule in successive turns of spiral. Beta plated sheet is formed by folding back of polypeptides. So in the next structure there is a tertiary structure. Tertiary structures contain of three types of bonds that are peptide, sorry, four types of bonds that are peptides, hydrogen bonds, disulfide bridges. Tertiary structure usually it is formed by bending and folding of different type of amino acid and forming a globular structure. So major is the that are the bonds that are present. There are four types of bonds that are present in tertiary structure, and in the secondary structure there are only two types of bonds, that one is the peptide and second is the hydrogen bond, and in the primary structure there are only one type of bond, that are the peptide bond present. So we can say tertiary structure are globular in nature, and if we add them into the aqueous media, then the hydrophobic region will be the inside the circle, and the hydrophilic region will be the on the surrounding, and this structure will be the more stable structure. Let's start with reading. Usually a polypeptide chain bends and folds upon itself forming a globular shape. This is protein tertiary conformation. It is maintained by three types of bond, namely ionic, hydrogen and disulfide, and peptide of course. For example, in aqueous environment the most stable tertiary conformation is that in which hydrophobic amino acids are buried inside while the hydrophilic amino acids are on the surface of the molecule. It is the most stable, just like a missile. And after that there is a quaternary structure in which there are also four type of bond are present, while there are no disulfide bridges that are present in the tertiary structure. There are hydrophobic interactions. Quaternary structure actually are the most complex structure, and these are formed by the aggregation, and there are peptide bond, hydrogen bond, ionic bond and hydrophobic. Most of protein of functional protein like hemoglobin is coordinated in nature. And start with the reading. In many highly complex protein, polypeptide tertiary chains are aggregated and held together by hydrophobic interaction, hydrogen bonding, and ionic bonding. The specific arrangement is the quaternary structure. Hemoglobin, the oxygen carrying protein of RBCs, exhibits such a structure. And this is the diagram of primary. You can see there is only direct linkage, that is the primary structure, and if there is a folding, some type of coiling, and you can see if there is a folding, it will be the secondary structure. You can see alpha helix, and after that there is aggregation due to the disulfide bridges, the it will be the tertiary structure, and after that the aggregation will be the quaternary structure. After that there are two types, there are classification of proteins. So it is very important long and short questions of classification of proteins. There are two types of protein. One is the fibrous protein and other is the globular proteins. Actually fibrous proteins contain the secondary structure more importantly, and globular proteins contain tertiary structure. You can see the difference and you can remember by comparing these two. Fibrous proteins are insoluble in aqueous media, while globular proteins are soluble in aqueous media. As the fibrous proteins are insoluble, so they cannot be crystallized. If one thing is insoluble in water, it cannot be crystallized, and these are elastic in nature. And the globular proteins are soluble in aqueous media and they can be crystallized. The next is very important, that is example: silk fiber that is obtained from the silk worm or spider webs, myosin that is present in the muscles, fibrin that is important for blood clotting, keratin that is present in nail and hairs. And the examples of the globular protein, that are examples: enzymes, antibodies, hormones, and hemoglobin. These are very important. Here globular proteins contain tertiary structure, importantly not always, because hemoglobin is quaternary structure in nature. So that was all about today's lecture. Thank you very much. And in the next lecture we will discuss about the nucleic acids, DNA and RNA, and the different types of RNA: transfer RNA, messenger RNA, and ribosomal RNA. Thank you. And in the next lecture we will discuss about the conjugated molecules.